So our next practical is subculturing of bacteria and fungi. So what is subculturing? The subculturing, it is a aseptic technique by which we can transfer the fully grown organism which is already grown on the solid state or any type of media. We have to transfer it into the freshly prepared media. That is the subculturing method. Okay, so we have to subculture the bacteria and fungi. So there are many methods of subculturing. The first one is the solid media to solid media. So when the organism is grown on nutrient agar plate, we have to transfer it. Okay, for it become alive. For that, we have to transfer it on the freshly prepared media. So that is the solid to solid. Suppose here is nutrient agar plate is there, okay, and the number of organisms are grown, okay. Then after some period, you have to transfer it into the new plate, which is freshly prepared media is there, or you can take the slants of nutrient agar. Okay, you can take these slants also as well as tab also. So how do you prepare in this subculture? First, the step is fully grown culture is necessary. If you have the fully grown culture, after that you have to transfer it into the nutrient agar plate or slant. So if there is a nutrient agar plate, then you have to follow the method that is strip plate method. Strip plate method, or you can call it as, there are two strip plate methods, zigzag and four corner, or you can, second one is spread plate method. Okay, by these two plate method you can subculture the given culture, that is solid to solid. And if you are thinking to transfer it into the on the slant, so just prepare the slant, nutrient agar slant, and stick on the nutrient agar slant. Okay, so what is the composition of nutrient agar? Composition. So peptone 0.5 gram, yeast extract 0.3 gram, beef extract 0.3 gram, agar agar, that is bacteriological powder, agar is necessary, that is 1.5 to 2 gram, NaCl, it is pinch of added, pinch of NaCl, and sixth one is distilled water, the distilled water should be 100 ml. Okay, so this is the composition of nutrient agar. We require peptone, yeast extract, beef extract, agar, NaCl. So I think all of you know about the raw materials, which means they are the, suppose the peptone is the source of nitrogen. Okay, agar powder is used for solidification. So all these materials and these raw materials and its sources, you should know it. Okay, and prepare this nutrient agar and add it into the test tube, sterile test tube necessary, and when you add it, tilt it at the 45 degrees angle and then wait for solidification. Your slant is ready now. Okay, once the slant is ready, then you have to strip on the slants. Okay, so this is the preparation of slant. And for stab, add only nutrient agar, sterile nutrient agar into the sterile test tube and wait for solidification. The stab is ready. Okay, so when the fully grown culture is provided to you, so you have to transfer it on the either the nutrient slant or stab or any type of petri plates. Okay, and then after the stripping, suppose here is the four corner method is performed. So after the streaking, you have to keep it into the incubator for incubation period. Okay, that is at the 37 degrees Celsius for 24 hours. You should keep this, and after that you observe the plates. There is a growth of organism, so that is the subculturing is done. Okay, and why we are performing the subculturing? To preserve the culture. Okay, when you are preserving the culture, at that time this culture should be transferred into the freshly prepared media. Okay, time to time. If you are doing this at on this week on Tuesday, so you should repeat this process for next Tuesday. Okay, why? Because the culture will be alive. You have to move it from one media, that is used media, to freshly prepared media after a fixed time period. That is the subculturing process. Okay. So students, for subculturing of the bacteria or fungi, so I prepared this nutrient agar. I told you the composition of nutrient agar. So I added all the ingredients in the nutrient agar, and then I sterilized all the material, that is media, as well as this petri plates, okay, along with the test tubes. See, the test tubes are also there. These are the test tubes, okay. So I will pour it into the test tube for preparing the slant, okay, as well as I will prepare the stab culture also, okay, slant, stab and plates, okay. So see students, I pour the nutrient agar into the test tube. Now I will tilt this test tube like this and I will wait for solidification, then it will become a slant, okay, and it is ready for subculturing. So see here how to add the nutrient agar into the plate. So within burners you should add this, okay. Again one more plate is there. So now we will wait for its solidification, okay. I will add and wait for solidification. So for subculturing of bacteria, see, the grown organisms are there on this slant, both slants, as well as on the petri plate also there is a growth of organism is there, okay. So I will take one colony from this and I will add on the freshly prepared nutrient agar. Okay, so see how I am adding, it is called as subculturing. So first all sterilize the nichrome wire loop very well, then take a colony from this. Okay, so I will take the colony. So one colony is there. It's too hot. Now I will stick on the plate or slant you can take. So this. I will prepare the 4-corner method. First corner is over. Now the second one. Subculturing on solid to solid. There may be solid to liquid is there, liquid to liquid is there, solid to liquid is there. Okay, means when you are taking the suspension or inoculum from the liquid, that is suspension, liquid media, then it is called as liquid to liquid. Okay, so this is the streaking is over. This is the solid to solid. Again I will show you the one colony. So this is, I will take the purple color colony on the agar plate and I will stick on the, wait for cooling. It is not seen. See here. Okay, so now I will take one colony purple color colony. Okay, and I will take a slant of nutrient agar and streak on that slant. Zigzag streaking on the nutrient agar slant. Why we are using the slant? Because slant gives the maximum surface area for the growth of organism, as well as it will provide the nutrients. Okay, so this is the subculturing method. So again, for fungus culture, I will take the fungal species from the plate and I will streak on the slant. Okay. So blackish fungus is there. So I will take the blackish fungus and I will streak on the nutrient agar plate. Okay. So I am taking. I already taken this culture. Okay, and I will add onto the slant like this. See, you have to streak like this. Zigzag, 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 zigzag. Okay, and then again close the and sterilize the nichrome valve. Okay, so this is all the slants are ready and I will incubate it, and after incubation period I will show you results. Okay, this is the subculture method.